Hey guys, this is our Lab 6 review video. I'd recommend checking out the one that Professor Braun gave us, but this is just the same material sort of condensed. And the purpose is easy to learn by teaching as well. So, all right, the layers of the skin, we have the epidermis, dermis, hypodermis. We'll go over the hair follicle a tiny bit, and then we have the different glands and sensor structures. Epidermis is the most superficial. It's come, let's get sunburn for stratum corneum, lucidum, granulosum, spinosum, and basal. The stratum lucidum is in thick skin only, and sometimes you can't see it. It's a glassy cell layer. Corneum is dead cells. Granulosum are cells that have granules to help aggregate keratin filaments, and so those are called keratohyalin granules. And then spinosum are several layers of keratinocytes producing keratin, and then our basal lamina is our meiotic cells with the intention that they're going to become melanocytes and then also keratinocytes. All right, so the cells of the epidermis, we have the keratinocytes, which make keratin, melanocytes, which produce melanin. They don't store it though. The keratinocytes take up the melatonin and store it. Then we have our Langerhans cells, which are immune cells, dendritic. They are phagocytic and they'll do antigen presenting after they eat the antigens. And then we have Merkel cells, which are acute touch sensing. In the dermis, we have two layers, the papillary layer, which is in contact with the epidermis. It's loose connective tissue, which is type 1 and type 3 collagen, and then it has elastic fibers. And then our second layer of the dermis is a reticular layer. It is dense connective tissue, has type 1 collagen only, and we have said it is unfortunately named because it doesn't have type 3 collagen. It does not have reticular fibers. Below that, we have our hypodermis, and that is our adipose tissue, has nerves, blood vessels, and the roots of our hair follicles. Also, our sensory the sensors and the glands are going to be there, with the exception of our Merkel cells, which are going to be in the epidermis. Okay, our hair follicles, we're going to go over a bit later, but we have the bulb, which has the core right here, and then we have the matrix, which is here, and the matrix is the part that grows. It's the keratin-producing cells that will grow and then form the keratin of the shaft of the hair. The shaft has an inner medulla, then the cortex, and then the cuticle on the outside, and then we have the internal root sheath and external root sheath, which are also made by the matrix cells down here. That are the ones that are doing the heavy lifting in terms of producing keratin. All right, and then we have our glands. So these are all in the hypodermis. The eccrine glands are the most common. They're the only ones of the three that doesn't excrete onto the hair follicles, which means that it can be in thin and thick skin. Eccrine secretes sweat via merocrine secretion, so just releases it onto the through the duct onto the surface. And then apocrine is pheromones and sweat in the genital region, anal region, and axilla, the armpits. Also merocrine secretion and not present on thick skin as it releases onto a hair follicle. And then sebaceous are associated with hair follicles, and so are the apocrine. They do holocrine secretion, which means the cells die as they release the sebum that they are excreting. All right, then we have our sensory structures. We already talked about the Merkel cells being the epidermis. We have the messeners, which are going to look like little Q-tips. They're in the papillary layer of the dermis. They're for low-frequency touch and twisted wool / Q-tip cotton appearance. I'm sorry, I said that these were all in the hypodermis. I stand corrected. Messeners corpuscle is in the papillary layer. Pacinian corpuscles are in the deep dermis / hypodermis, and those are for pressure and vibration. All right, but messener is low-frequency touch. Pacinian is for deep pressure and vibration, and then Merkel is for acute touch on the epidermis. All right, parts of the vascular system we're going to be taking a look at. I did make a video on this, so you can watch that, but basically we're looking at the three layers of the connective tissue around the vessels. We have the innermost tunica intima, then the intermediate layer, the tunica media, and then finally the tunica adventitia. And the idea is that the bigger vessels are going to have all three. The capillaries are only going to have the tunica intima, and then we have the arterioles and the postcapillary venules. The metaarterioles and the postcapillary venules are going to have tunica intima only. But the presence of these guys and what they look like is going to allow us to distinguish different types of vessels. All right, so what are the different types of vessels? Well, we know we have arteries, veins, capillaries. Arteries, we have elastic arteries, muscular arteries, arterioles, and metaarterioles. Capillaries, continuous, ventral, discontinuous veins, postcapillary venules, muscular venules, and medium to large veins. All right, so we're going to go through all these in just a minute. So I'm not going to go into detail, but you're welcome to watch the other video. Other thing to know, we have something called AV shunts. So AV shunts are arteriovenous shunts, direct arterial to venal connection to bypass capillary beds. And then our portal systems, we have arterial and venous portals, arterial in the renal cortex and venous in the hepatic and pituitary glands. It's when two capillary beds are in series, connected by an arterial or connected by a venule. All right, then we have, so looking at our thick skin, we have the hypodermis region, the dermis region, and then the epidermis. How do we know that this is thick skin? Well, this is exceptionally thick. We also don't have any hair follicles that are obvious. And then one other thing to notice, the hypodermis is going to have our subcutaneous fat, so you can see a whole lot of fat, and that's a good sign that you're in the hypodermis. All right, layers of the epidermis. We like our mnemonic, come, let's get sunburned. So here we have our superficial part here, and then deep is here. So 4 is our stratum corneum. 3 is going to be our stratum granulosum, and then right above it would be our stratum lucidum, but it's hard to tell in this picture. What it would be is like glassy pink, but in this particular instance, you don't really see it here. This is thick skin though, so stratum lucidum would appear in thick skin, does not appear in thin skin. All right, then we have the stratum spinosum. It makes keratins. If you were to zoom in, you'd be able to see tiny little spines between the cells, and those are remnants of desmosomes adhering the cells together. And then our final layers are stratum basal, which is mitotic to make the eventually the stratum corneum, right? So the idea is you have mitosis here to make all of the cells in the layers above it. Okay, deep to the stratum basal, we have the dermis, and so here we have the papillary layer, and then here we see a lot of dense connective tissue is the reticular layer. Over here we have additional parts of the papillary layer, and it's just been cut at a weird angle, so it's hard to tell that it's papillary because it looks like it's embedded in the the stratum spinosum, but is in fact not. It is part of the papillary layer. It's just cut strangely. All right, and then remember the papillary layer has these papillae, these protrusions into the epidermis, and that's to help keep the dermis and epidermis adhered to each other. All right, sensory structures of the skin. So we said that the Messinger's corpuscles are going to look like a Q-tip. In particular, it kind of looks like the cotton fibers wrapping around a stick. So you can see we have a whole bunch of nuclei on the outside, and so it kind of looks like you were just trace those wrapping around the center. And so what that is the center stick part is the axon of a cell, right? Because this is a sensory structure. It's going to have a nerve in it, so or the axon of a nerve. And then these are all Schwann cells. We remember Schwann are myelinating cells in peripheral nervous system, which were in the skin here, so this must be peripheral. And then oligodendrocytes are myelinating cells in the central nervous system, which we're not. So those must be Schwann cells on the outside right there. But this is the Messenger corpuscle, and it's light touch and low frequency vibration. And we can see that it is in the dermal papillae, part of the dermal layer. Again, what's going on? It's at a weird cut, but it would look more something like this. It's at a cut, so it looks like that. Right? Okay, weird cut angle. Then we have the Pacinian corpuscle. This is deep pressure sensor for mechanical and vibratory pressure. These ones are really easy because they look like onions. All right, so Pacinian corpuscle, Pac-Man. He likes to eat fruit, but I bet you he would also like to eat an onion. All right, so deep pressure. All right, then we have our thin skin. How can we tell this is thin skin? Well, this is the epidermis right here. You can see that entire layer is right there. It is much thinner than the thick skin we saw before. Okay, so we have 1 is the epidermis, 2 is the dermis, and then 3 is our hypodermis. This is part of the scalp. We can start to pick out parts of so this is like stratum basal. Come, let's get sunburned. So stratum spinosum. Get this right here would be our stratum granulosum. And then so there's going to be no lucidum. So that means this is stratum corneum. All right, and then we're told to look for brown material. This brown material here, you're like, oh, that must be our melanocyte. That brown material is in fact melanin, but it's being held by keratinocytes. So this is not a melanocyte. This guy right here and this guy with the clear cytoplasm are our melanocytes, right? But the ones with the brown in it, those are the keratinocytes holding on to the melanin that the melanocytes made. All right, it's the exact same thing. Melanocyte, clear cytoplasm. It is not holding on to the melanin it's making. The melanin, brown substance, is accumulating in the keratinocytes. All right, then we have our hair follicles. This right here, so this is our bulb of our hair. The first part right here is our hair papilla, and this is going to be loose connective tissue. Then outside of it, in this area, we have our hair matrix. And this is the meiotic layer that is going to do division and produce keratinocytes, and its divisions are going to produce the external root sheath, internal root sheath, and the keratin that makes is going to form the shaft of which you can start to see here, the shaft of the hair follicle. All right, so we're going to start to see that here. Right here is the shaft of a hair follicle. This is the internal root sheath. This is the external root sheath, and then we have connective tissue right there. But we notice that the external root sheath has tons of cells. The internal root sheath is thin, and the hair shaft is fairly light, poorly stained. And then the connective tissue sheath, external to everything, is just that, external to everything. All right, now we have our sebaceous gland. Our sebaceous gland is located right here. What's a good hint that it's a sebaceous gland? Well, it is right next to our hair follicle. And so you can see that, there's no like very clear lumen. It seems to kind of lead up to maybe the hair shaft right here with a weird cut. But it kind of just looks like a blob overall. And it's going to release sebum via holocrine excretion, which we talked about already, on the shaft of the follicle. Let's go to the surface. Then we have different parts of the hair follicle again. External root sheath has lots of cells. Again, this is coming from the matrix. Then we have the connective tissue outside of that. All right, and then we actually have we can start to see it, but right here and up here, there's some smooth muscle, and that's going to be our erector pili muscle, which is what allows your hair follicles to stand on end when they're receiving their stimulation. All right, so then we have our hair follicle cross-section, and again, easiest one to find is the external root sheath. Why? It's dark, lots of cells. Our internal root sheath is relatively thin, not as dark, right? And then on the inside, we have our nice hair shaft. All right, so it's our hair shaft. And then our connective tissue sheath is on the outside, relatively thin, and pink staining. All right, then we have our other glands. So it looks like we actually have all 3 of our glands on this page, but we'll start with the first one, which is our eccrine sweat gland, which I'm sure I mispronounced. Right here, this is our, we have 2 parts right here, the light stained portion of it, and then the dark stained portion. So if it is looking something like this, right, the dark stained portion is going to be the duct portion, and then the light stained portion is where the cells that are doing the secretion are located. So lighter stained, we're cut further towards the secretory portion. Darker stained is towards the top of the gland, the duct of the gland. All right, but the inside of the eccrine sweat is stratified cuboidal. We can also see some here. Those are dark, so that's going to be the duct of the glands. All right, then over here we have the apocrine sweat gland, and so that tells us that this must be we have to be in thin skin, right? Because we have a hair follicle here, so that's thin skin. But we also know that apocrine is going to be basically produce their pheromones and their sweat onto the shaft of the hair follicle and then take that to the surface. And so this is only in the axilla and anal genital regions. They have simple cuboidal or columnar, and then basically it's huge lumens, right? That's how we know. So big lumens for apocrine. All right, and then our third one is down here, and this is our sebaceous glands, and we can see that this appears to be the shaft, and they're the shaft of a hair follicle. But again, it kind of looks like a blob, and we don't have these lumens that are so obvious in apocrine or the eccrine sweat glands. All right, so again, sebaceous glands, and then we have our internal root sheath here. B is our external root sheath. Look at all those cells. D is the hair follicle itself, and then C is the external part. All right, now let's take a look at our blood vascular system. Here is an elastic artery. It's stained with resorcin fusion, which is going to pick up those elastic fibers. And then we have the tunica intima, tunica media, and tunica adventitia. Tunica media, intima, excuse me, is this one. And that's going to be lined by an epithelium right here. It's the vascular endothelium, and it's simple squamous. We're going to have collagen and some elastic fibers. If you look at it closely, elastic fibers would be not a complete lamellae, not a complete ring. But then you can start to see that we have our first one right here. And so this one is going to be our internal elastic lamellae, and that marks the end of the tunica intima. And that brings us to the next one, which is tunica media, and that one likewise also ends in our elastic lamellae, and this is the external elastic lamellae right here, and again circular ring lamellae of elastic fiber. And that brings us to our last one, which is tunica adventitia. And one of the things to know is that, or to note, the tunica intima is all sort of like this, right, wrapping around the lumen. Same with the media, but the lamina, it's not so clear, right? You could have weird cross section, but it's not all wrapping around the lumen. All right, then we have muscular arteries and veins. So this is not a muscular vein. This is just a vein, right? And then this is a muscular artery. How do we know that's the case? Well, we have, starting from the innermost and working our way out, this is this black line here, darkly stained, because it's stained with resorcin fusion. That is a very darkly stained internal elastic lamina, and that's going to mark the boundary of our tunica intima. And so everything past that is going to be our tunica media, right? And so this tunica media is going to have, you can see that it has a whole lot, it's very robust, strong tunica media, strong round shape. It has 3 to 40 layers of smooth muscle, right? This one has around 5, but you can tell it's like robust, round shape, very much more than 1 to 2 layers of smooth muscle. So therefore, that must be a muscular artery. All right, but compared to the elastic artery, where, right, do we see smooth muscle here? No, we see a bunch of elastic layers, and so that is an elastic artery. This one has got a bunch of smooth muscle. This is a muscular artery. All right, and then outside to that, we have our tunica adventitia, and you can see again these all appear to wrap around. This is a little bit perpendicular almost to the tunica media below it. Right, it doesn't quite look like it wraps around the inner part, the lumen, the same way. All right, then we have our vein, and you can see that the vein has a very, very prominent tunica adventitia. It has less integrity, right? It's kind of a weird shape. The lumen is quite weird. And then it's important to remember that arteries and veins go together. So this guy is very clear what this one is. Muscular, it's got to be an artery. It's keeping its shape. And then the vein that's associated with it doesn't keep its shape very well. It's got a huge tunica adventitia. All right, then we have our arteriole and a muscular venule, and the resolution on this is awful, but we know this is a venule, or arteriole, because it does look a lot like the muscular artery, but there's only 1 to 2 layers of smooth muscle. All right, so this tells us this is an arteriole. It does have the internal elastic lamina. You can see this black line, but sometimes they don't have this. Really, the key is that 1 to 2 layers of smooth muscle, which we can see here. All right, there's also some tunica adventitia on the outside, but without an elastic stain, it would be harder to see. And then this guy right here, very hard to tell at this resolution, but this is a muscular venule. How do we know that? It's got 1 to 2 layers of smooth muscle. Right. All right. Let's move on. So finally, we have our vena cava, which we know is going to lead back to the heart. It's our big vein. Here we can see we have our tunica intima is right here. Our tunica media is right after that. You can see the internal elastic lamina with these small arrows right here. And then, again, notice that you could appear to draw like a line along the entire thing. So it would appear that the tunica media and the tunica intima are like wrapping around that lumen. But here, it's just less clear. The lines are all kind of broken up. And so it looks like we might have some objects that are, or some parts that are not quite as concentric, right? And so it's a good sign that we're in the tunica adventitia. It does have, smooth muscle, but again, not wrapped around in the same way. So that's the vena cava. All right, we're almost done. This is our electron microscopy. We said we had 3 types of capillary: continuous, fenestrated, and discontinuous. So this one, again, it's hard to see these in light microscopy unless there's a red blood cell inside of it. Why? Well, like the red blood cell is basically the size of the capillary, right? It's filling up the entire thing. But notice how there's, we can see some tight junctions here, but there's no clear holes, right? So this is continuous. Then we have fenestrated. You can see that there's like little beads on the edge, very thin, and so those would be our fenestrate, or like the tiny holes. But you're not to have red blood cells go through those because they're not particularly big holes. So like small things can go through, proteins, etc., but not the big red blood cells. And then you have discontinuous, and you can see that we have these big chunks of missing endothelium. And so in this case, you would have a discontinuous capillary. You could theoretically have red blood cells slip through those. All right, so that's the end of the lab. We have some review questions. We'll try to go through those very quickly. All right, so what is this? Is this thick skin or thin skin? This is thin skin. Why do we know that? We have a hair follicle. This must be thin skin. Also, the epidermis appears to not be very thick, even though we're zoomed out. All right, so where is the epidermis? Epidermis is right here. Where's the dermis? That would appear to be this region 2. So 3, 2, and then 1. 1 is our hypodermis. All right, integumentary, label the parts of the skin. All right, well, so this one, epidermis. B is dermis. A is hypodermis. All right, then we're going to label the glands: sebaceous, hair follicle, and eccrine. All right, well, I think that this right here is probably a cross-section of a hair follicle. So 2 is going to be our sebaceous, right? It's always next to a hair follicle. Then we have one is going to be our eccrine, right? We can see the duct of the eccrine, and then some of this lighter staining stuff is the secretory part. So 3 is hair follicle, and 1 is eccrine. All right, different part, we have epidermis again. This appears to be thin skin as well. Come, let's get sunburned. So come, this is going to be stratum corneum. I don't know why my iPad's doing that. All right, so that's this guy. Then we have, this is thin skin, so let's is not here, so there's no stratum lucidum. Come, let's get, so this is going to be our stratum granulosum. Burned, that's the stratum, come, let's get sun, stratum spinosum. All right, and then that's going to have our keratinocytes in it, producing keratin. And then let's get sunburned, stratum basal, which is our mitotic layer. All right, then we have here, this appears to be the part of the dermis, and it's adjacent to the epidermis. This is our papillary layer. And then this, hard to tell, but it does appear to be circulating a melanocyte. And then 2 is referring to this guy. Again, kind of an uncool question, because I think that, like, what the heck, that is a continuous capillary. All right, how would you know that? Well, you can see that the diameter is approximately cell-sized, and there's no obvious gaps. Of course, it's very low resolution, so I didn't think that's a fair question. All right, 3 glands. Well, this is not too bad. This one is a weird blob, and it's next to a hair follicle, so that tells us that this one is sebaceous. Then we have these guys over here with huge lumens, so that must be apocrine. And then, last but not least, this guy over here, with the dark parts being the ducts, and then the lighter parts being the secretory parts. Those are eccrine sweat glands. All right, vascular system, label the following. All right, well, so this is nerve tissue. How do we know? It looks kind of foamy. All right, so that's nerve tissue. Ooh, these are going to be very hard because it's very small. Let's see, what, maybe we can find out what G is. G looks like muscle, so I'm thinking this is G. All right, and then they are so small and it's such a low resolution. Yeah, hard to tell. All right, well, we can make some educated guesses. F appears to be more circular, A appears to be more circular. So I'm going to say that those are both or some form of artery. B appears to be bigger, so I'm guessing this is going to be B, or muscular venule, and muscular artery, I think, yeah, it's very hard to tell. Too small. All right, we give up on that one. But I'm sure we can tell if we get a better picture. All right, last, electron microscopy and light microscopy. Arterial, so this is just exactly that. In arterial, we can see that it's got layers of smooth muscle, right? Arterioles are 1 to 2 layers of smooth muscle. And then we have, yeah, so here's 1 layer of smooth muscle. Here's another part. And it's circular, which tells us that, you know, it's probably going to be some sort of artery. So arterial, it's not muscular artery because we have only 1 layer or so of smooth muscle. We also know it's not a capillary because capillary, basically 1 cell would take up the entire cross-section of the capillary. So if it's not a capillary, it's not muscular. We don't see a ton of layers of elastic lamellae, so it's not elastic. So that pretty much leaves us with arterial. So process elimination can guess that. Here's another arterial, very small, I think 1 to 2 layers of elastic, or 1 to 2 layers of smooth muscle there. All right, so that's it. Good job, and we're done with lab 6.